Usually when we talk about a medical diagnosis, there is this expectation of precision, like engineering almost.
Yeah.
You break your arm, the X-ray shows that jagged white line, and the doctor just points and says, there it is.
It's comforting, because it's visible. You can pinpoint the exact location of the hardware failure.
Broken or not broken. But then you step into the world of neurodevelopment and aging, and suddenly that X-ray machine is totally useless.
No, completely.
We have this universal fear of getting older and losing our memories, and we almost always blame it on the brain just wearing out, like an old engine.
Exactly. That's the default assumption.
But what if the brain isn't actually the primary culprit? What if it's just a perfectly healthy engine getting a bad signal?
From the stomach, of all places.
Right. So welcome to the deep dive. Today, we are ripping up that traditional X-ray. We're unpacking some wild, groundbreaking research out of a 2026 Nature paper.
And it is a paradigm shift, for sure.
Our mission today is to map out this newly discovered biological chain reaction that causes age-related memory loss. And it does not start in the brain.
It really doesn't.
It starts in your gut, travels up a nerve, and literally shuts down your brain's memory centers.
To really grasp how this happens, we need to look at a core concept called interoception.
Interoception, right.
Yeah. We all know our external senses: sight, sound, touch. Interoception is your brain's ability to perceive the internal state of your own body.
Like knowing you're hungry or feeling your heart racing.
Exactly, or knowing that you're fighting an infection. This paper proves that when that interoceptive connection between the gut and the brain breaks down, your memory encoding just fails.
It's wild. So we're going to trace this dysfunction step by step. And the whole thing kicks off with what I like to call a bad roommate situation in the gut microbiome.
That's a great way to put it.
The researchers asked, does an aging gut actually cause cognitive decline, or is it just a coincidence?
Right. Correlation versus causation.
Exactly. To figure this out, they did something kind of brilliant, but also a little gross. They took young mice, just 2 months old, and co-housed them with elderly 18-month-old mice.
The laboratory setup here is crucial. Mice are naturally coprophagic.
Which means they eat each other's feces.
Yes. While that sounds unpleasant to us, in a lab, it's a natural way to transfer gut bacteria.
Right.
If you put young and old mice in the same cage, those young mice will very rapidly acquire the gut microbiome of their older roommates.
So within a month, these young mice effectively have an old microbiome.
Yes.
And the behavioral results were staggering. The young mice developed severe short-term memory loss.
They really did.
They tested them using a novel object recognition task, which is basically seeing if a mouse remembers a little plastic toy it's seen before, and a spatial memory test called the Barnes maze.
And they just completely bombed these tests.
But of course, rigorous science requires ruling out alternative explanation.
Right, because a skeptic would say maybe the young mice were just stressed from living with older, grumpy mice.
And we know chronic stress can impair memory.
Exactly. Which is why the next phase is so elegant. To rule out social stress, they performed a fetal microbiota transplant, or FMT.
Okay, so extracting the microbiome from old mice and transplanting it into young, germ-free mice.
Right. And these mice lived entirely alone in sterile environments. No roommates to stress them out.
And the outcome was identical. Their memory plummeted.
But what's really fascinating is what didn't change. Their exploration behavior, their curiosity, their physical strength.
It all remained completely normal, right?
Yes, it didn't make them frail or depressed. It acted like a laser, specifically targeting their memory.
And then the researchers hit them with the ultimate undo button. They gave these mice a 2-week blast of broad-spectrum antibiotics.
Like neomycin and ampicillin.
Yeah, just to wipe out the gut bacteria. And it completely restored the memory of the young mice. But wait, it also improved the memory of the naturally aged mice.
That finding upends everything.
If a simple round of antibiotics can fix the memory of naturally old mice, it means memory loss isn't a permanent hardware issue.
Exactly. The neural architecture for memory is intact. It's a software glitch caused by bacteria.
So they sequenced the bacteria and isolated one specific strain, Parabacteroides goldsteinii.
Which naturally explodes in population as we age.
And when they introduced just this one bacteria to germ-free mice, it single-handedly wiped out their memory.
It's the primary instigator.
Okay, let's lock this in with a mnemonic I came up with for you guys listening. It's GMIVH. Great memories intentionally vanish here.
Oh, I like that.
G is for gut bacteria. M is for metabolites. I is for immune cells. V is for vagus nerve. H is for hippocampus. We just covered the G.
For bacteria.
So let's do our first quick recap. Bullet 1.
Yeah.
Aging changes the gut microbiome, specifically increasing Parabacteroides, goldsteinii.
Yes.
Bullet 2, transferring an old microbiome to a young body transfers memory loss. Bullet 3, this memory loss is completely reversible with antibiotics.
Spot on.
So a question for you listening. Think about your own gut health. How often do we consider that a stomach ache might be directly altering our ability to remember where we put our keys?
It's a very real possibility.
But that brings up a massive physical disconnect, because a bacterium sitting in your intestines isn't traveling up your spine to your brain.
No, it doesn't travel at all.
So how does it reach the skull?
It acts from a distance using chemical messengers. As this bacteria ferments food, it secretes specific metabolites.
This is the M in our mnemonic, metabolites.
Right. Specifically, medium-chain fatty acids, or MCFAs. The main troublemaker is a chemical called 3-HOA.
I love how they prove this part. They took the liquid culture the bacteria were growing in and they rigorously filtered out every single bacterial cell.
Filtered down to less than 3 kilodaltons, just pure liquid.
Right, so just the chemical soup. They fed that to the mice, and the mice still lost their memory.
Confirming the damage is done by the secreted chemicals, not the physical bacteria. Now, visualize these MCFAs as billions of tiny chemical keys floating around the gut lining. A key is useless unless it finds a lock.
And they found the exact lock, a receptor called GPR84.
Yes, and location is everything here.
Where exactly is this lock found?
Almost exclusively on peripheral myeloid immune cells in the gut. Macrophages, monocytes, neutrophils.
So the local immune system.
Exactly. They constantly patrol the intestinal wall.
So if the bacteria are like arsonists, these MCFAs are the chemical matches they're tossing around.
Like that analogy.
And the immune cells are the dry brush. When the matches hit the GPR84 receptor, the brush catches fire. The immune cells panic and flood the gut with inflammatory cytokines.
Specifically interleukin-1 beta, or IL-1 beta, and TNF.
The evidence here is incredibly tight. They looked at DBA mice, which have a natural mutation where they lack the GPR84 receptor.
They simply don't have the lock.
Right. And when researchers flooded these mutated mice with the MCFAs, nothing happened. Memory stayed perfectly intact.
Because the chemical key had nowhere to turn.
And they double-checked this by using drugs like PLX33927 to deplete those immune cells.
Or neutralizing the IL-1 beta directly.
In every scenario, if you block the immune panic attack, you protect the brain, even if the bacteria is still there.
The inflammation is the mandatory middleman.
But let me push back on this. We hear about inflammation all the time. Inflammaging, right?
Yes, systemic inflammation as we age.
So why does this specific gut inflammation cause a highly targeted issue like memory loss? And not just, a stomach ache?
It's an excellent question. The key is what the inflammation is physically touching. It's surrounding the endings of the vagus nerve.
Ah, the vagus nerve. The V in our GMIVH mnemonic.
Yes. The inflammation is blinding the communication wire to the brain.
Okay, time for our second quick recap. Bullet 1. The bacteria release medium-chain fatty acids like 3-HOA. Bullet 2. These MCFAs activate the GPR84 receptor on gut immune cells. Bullet 3. Immune cells release inflammatory cytokines like IL-1 beta. So a question for you listening. If gut inflammation is the middleman for cognitive decline, could the anti-inflammatory foods you eat actually be protecting your brain's hardware?
It certainly provides a hard biological mechanism for that.
Okay, so the fire is raging in the gut, and it's engulfing the vagus nerve. Let's talk about that wire.
The vagus nerve is the superhighway of interoception. It sends constant, high-speed sensory data from the gut directly into the brainstem.
And the live imaging experiment they did here is mind-blowing.
The calcium imaging, yes.
Right. They looked at a cluster of vagus nerve cells called the nodose ganglion. When they pumped nutrients into young, healthy mice, that vagus nerve lit up like a fiber optic cable.
Bright and robust.
But in the mice with the old microbiome, the signal was incredibly weak.
Almost entirely muffled. And we know why. That IL-1 beta physically suppresses those specific vagal sensory neurons.
So it's not destroying the nerve cells?
No, it just chemically dials their volume down to near zero.
It's like a transatlantic telephone cable. The brain is waiting for a daily status report to know everything is okay downstairs.
Right.
But the inflammation creates so much static on the line that the brain only hears dead air. But wait, why does the brain care?
What do you mean?
If I'm trying to remember a phone number, Why does my brain need a status update from my stomach to form that memory?
That is arguably the most profound insight of this entire paper. The brain is an extreme energy management system. It only accounts for 2% of your body weight, but consumes 20% of your energy.
Right, it's a gas guzzler.
Highly expensive. So forming a new permanent memory and enneagram takes a lot of energy.
So the hippocampus needs reassurance that the body is fueled and stable before it commits that energy.
Exactly. It relies on continuous baseline signals from the vagus nerve. If that input drops out due to inflammatory static, the brain interprets that as a system-wide warning.
Wow.
To conserve energy, the memory centers basically drop into a low-power sleep mode. No signal from the gut means no memory encoding.
It's a built-in safety mechanism. Halts non-essential functions.
Precisely.
Okay, recap time. Bullet 1. The vagus nerve senses the gut and reports back to the brain.
Yes.
Bullet 2. Immune inflammation physically suppresses vagal nerve firing. Bullet 3. The brain receives a weakened body status signal.
Perfect summary.
So to the listener, have you ever eaten a massive unhealthy meal and felt that dense brain fog an hour later?
Oh, the classic food coma.
Right. Could you literally be feeling the temporary inflammatory static on your own vagus nerve?
You almost certainly are. It's an acute version of what these aging mice experience chronically.
Incredible. Okay, so the signal has dropped. Let's look inside the brain itself. The H in our mnemonic hippocampus. What does this low-power sleep mode actually look like?
Researchers looked at the expression of an immediate early gene called FOS. FOS.
I love the FOS gene. It acts like a microscopic light bulb inside a neuron.
That's a great analogy.
It flashes on precisely at the moment a neuron is actively encoding a new memory. It's the biological equivalent of the brain save button.
Yes, and when healthy young mice see a novel object, their hippocampus lights up with FOS expression.
The save button is being pressed.
Exactly. But in the older mice, or the young mice with old microbiomes, the hippocampus remained totally dark.
The save button was mechanically locked out.
The FOS response was severely blunted.
But, and I think this is the most hopeful finding in the study, they proved it wasn't a structural loss.
Right. They checked the dendritic spines, the physical connections, perfectly fine.
They checked for neurogenesis, the birth of new neurons. Totally normal.
The hardware was perfectly intact. It just wasn't being permitted to turn on because the green light from the vagus nerve was missing.
So the brain isn't fundamentally broken or rotting away.
Right.
It's just waiting for an all clear from the gut that never arrives.
It shifts cognitive decline from a narrative of inevitable decay to one of a highly treatable communication dysfunction.
That is massive. Okay, recap number 4. Bullet 1. The hippocampus requires vagal input to encode memories. Bullet 2. Without the signal, FOS activation drops, memories aren't saved. Bullet 3, the physical brain structure is completely intact.
A functional decline, not a structural one.
Right. So a question for you listening. If your brain's hardware is actually fine, how does that change the way you view aging?
It opens up an entirely new world of therapeutic targets.
Which brings us to the final part, the interventions. The algorithm of rescue.
Because it's a multi-step pathway, they found they could intervene at almost every single step.
Let's walk through the toolkit. Step 1, target the bacteria directly.
They used phage therapy, a specific virus called Bacteriophage 5-PDS1 that hunted and killed only the Parabacteroides.
And memory improved.
Yep.
Step 2, target the immune receptor.
Right, using drugs to block the GPR84 lock, no inflammation, memory rescue.
And 3 is where it gets really wild, bypassing the gut entirely and targeting the vagus nerve.
They used a low dose of capsaicin.
Wait, capsaicin, the exact chemical that makes chili peppers spicy?
The very same. Capsaicin is a potent activator of TRPV1 receptors on vagal sensory neurons.
Oh, wow.
By administering it, they forcefully jump-started the vagus nerve. It shouted over the inflammatory static.
And memory was instantly restored. It's like finding a manual override switch in a hot pepper.
It really is.
But there's a 4th step that connects to something millions of people are experiencing right now. Target gut hormones.
Yes. They use CCK and GLP-1 agonists, specifically the drug liraglutide.
Wait, liraglutide is a diabetes and weight loss medication.
Exactly. It naturally stimulates the vagus nerve. When given to old mice, memory returned to normal youthful levels.
We hear anecdotal reports from patients on these modern GLP-1 weight loss drugs saying their brain fog suddenly lifted.
Well, this provides a mechanistic explanation. The researchers coined a term for this: Interoceptomimetics.
Interoceptomimetics.
Compounds that mimic healthy body-to-brain signals. They forcefully stimulate that communication line, tricking the brain into feeling optimal bodily health.
So it hits the save button again, even if the underlying aging is still there.
Yes, counteracting cognitive decline entirely from the periphery.
Okay, final recap of our solutions. Bullet 1. Precision phage therapy can eliminate the bacteria.
Right.
Bullet 2. Capsaicin can manually jumpstart the vagus nerve. Bullet 3. Gut hormone drugs like GLP-1 act as interoceptomimetics.
Beautifully summarized.
So, knowing that compounds like capsaicin or GLP-1 can manually override the aging gut, what future therapies do you think will emerge?
I think we'll see a pivot. Cognitive therapies that don't even try to cross the blood-brain barrier. They'll just focus on optimizing the gut and peripheral nervous system.
It's all connected. Let's tie the whole GMI-VAH cascade together one last time. Aging allows a specific bacteria to bloom.
They make the chemical matches, the MCFAs.
The gut immune system freaks out, starts a fire. The vagus nerve goes numb from the static.
And the hippocampus fails to hit save.
The paradigm shift here is profound. Brain aging is heavily driven by brain extrinsic factors.
It's not just about what happens in your skull, it's about the signals your body is sending.
Exactly. So I want to leave you, the listener, with a final provocative thought to mull over.
Lay it on us.
If our memories depend entirely on the brain constantly feeling the health of the body, does that mean cognitive decline isn't a disease of a broken brain, but actually a disease of sensory isolation?
Oh, that's a fascinating way to frame it.
Right. If we can find ways to keep the body talking loudly to the brain, could we theoretically keep our memories sharp forever? Something to think about. Until next time.